Now we will take a look at the cyclic form of monosaccharides. For 5 or more carbon atoms, the cyclic structure will dominate at equilibrium with the open chain structures. So before we talk about the cyclic form, we need to review one important reaction between a carbonyl of the aldehyde or ketone with alcohols. Do you still remember this reaction? Alcohol is the nucleophile. It will add to the carbonyl carbon to form this hemiacetal, right? It's a nucleophilic addition of alcohol to the carbonyl carbon. The cyclic form for monosaccharides are actually hemiacetal. So let's first take a look at the reaction between the OH group and the carbonyl within the same molecule. So the structure shown here is 4-hydroxybutanol. We have OH on carbon 4 of this aldehyde. So an intramolecular nucleophilic addition will take place. The OH on carbon 4 will serve as the alcohol to attack on the carbon 1 carbonyl. As a result, a ring structure will form, and this ring will involve carbon 1, 2, 3, 4, and the oxygen on carbon 4. So 5 atoms are involved in the ring, and it's going to be a 5-membered ring form. All right. The carbon 1 here, carbon 1 was the aldehyde carbonyl, right? In the product, carbon 1 has an OH attached to it. This OH is from the opening of the C double bond O. Okay, CO double bond opens, it gives an OH. And the ring oxygen is derived from the hydroxy group on carbon 4. All right, so this is a cyclic hemiacetal derived from the intramolecular nucleophilic addition of the OH to the carbonyl carbon. And there is a very specific term for carbon 1 here. We call it anomeric carbon. Anomeric carbon refers to the carbon that has 2 oxygen atoms attached to. Okay, anomeric carbon is the carbon that's derived from the carbonyl carbon. All right, let's see one more example. Here we have 5-hydroxypentanol. When the intramolecular nucleophilic addition to the carbonyl takes place, it's going to form a ring. And what kind of ring will we have this time? How many atoms are involved in the ring? Carbon 1, 2, 3, 4, 5, as well as the oxygen attached to carbon 5, right? Because it's this oxygen that's going to make a bond with carbon 1. So 6 atoms are involved. It's going to form a 6-membered ring. And the carbon 1 is the anomeric carbon. This is the carbon that's derived from the carbonyl carbon. And this is the carbon that's attached to 2 oxygen atoms in this cyclic hemiacetal. All right, Now we have some specific terms to introduce in the carbohydrate chemistry. If we have a 6-membered ring cyclic hemiacetal for a sugar molecule, we will call it pyranose. So pyranose means the sugar molecule forms a 6-membered ring hemiacetal. Okay, the cyclic hemiacetal is a 6-membered ring, pyranose. And similarly, for the 5-membered ring, we call it furanose. Okay, 5-membered ring, furanose. All right, so we would include this pyranose or furanose in the names of the cyclic sugar, and so you know what kind of ring it will involve. All right, so now let's take a look at how to convert the open chain Fischer projection to the cyclic form for monosaccharides. Because the monosaccharides contains so many stereocenters, one of the key is to keep all the stereocenters the same, because the stereocenters are not affected, right? So I want you to follow what I'm going to present here to do this systematically. Let's use D-ribose as an example. Okay, so the first step you are going to do, you want to lay the molecule down. In Fischer projection, it's vertical. You want to lay the molecule down. And you want to put your carbon 1 on the right-hand side. Okay, carbon 1 on the right-hand side. And in this process, all the OH groups on the right-hand side in the Fischer projection will be pointing down, and all the OH to the left side will be above, right? Now, second step, we are going to curl the carbon chain up, which means we are going to put the 2 terminal carbons, 1 and 5, up, or curl the molecule up. So this is what I mean. In this process, you do not want to change any stereocenters. All the OH that are pointing down will still be pointing down. We just curl the molecule up. All right. Next, now I want the carbon 4 OH to react with carbon 1 aldehyde. Okay, carbon 4 OH react with carbon 1 aldehyde. Where is carbon 4 OH now? It's pointing down, right? So it's not close to carbon 1. So we need to rotate a bond to make carbon 4 OH close to carbon 1. So what bond can we rotate here? The C3C4 bond. So we are going to rotate the C3C4 bond so that the C4OH will be pointing to the right-hand side. And as we rotate this bond, C4OH to the right-hand side, then the C5 CH2OH, this part, will now be pointing up, right? All right, so now the C4OH is close to carbon 1, and the nucleophilic addition to carbonyl can take place. And this is how the ring is formed. And in this case, it's going to form a 5-membered ring, right? Again, keep the stereocenters. The stereocenters will not change. But what about the carbon 1? Carbon 1, previously it's an aldehyde carbon. It's not chiral. But once we form the cyclic hemiacetal, we actually generate a new chiral carbon here on carbon 1. So on carbon 1, we have an OH group. This OH group could be either up or down. It has two possibilities, right? But the rest of the stereocenters will stay the same, right? So we call these two forms as alpha and beta. Okay, one is in the alpha form, one is in the beta form. How do we define alpha and beta? Well, let's see the two structures one more time again here. These two structures has only one chiral center different. All the other chiral centers are the same. There are 4 total chiral centers, right? So they are diastereomers of each other, or you can also call them epimers. Remember, we talk about epimers. Epimers are those with many stereocenters, but only one center has the opposite configuration. The rest are the same. So these two are also epimers on carbon 1. And the first structure, we can call it beta D ribose. The second one, alpha D ribose. Whenever you see alpha or beta as prefixes, you know this refers to the cyclic form. All right. So what is alpha? When the OH is on the opposite side of CH2OH on carbon 4, we call it alpha. So this term is all relative. Okay, relatively speaking, carbon 1 OH is on the opposite side of the CH2OH on the last carbon. We say this is the alpha form. And beta, when the OH is on the same side as the CH2OH on the last chiral carbon. Okay? And now we have 5-membered rings, hemiacetal. And we just mentioned that 5-membered rings, sugar molecule, we also call them furanols, right? So the complete name for these two structures, the first one will be beta D ribofuranols, and the second one will be alpha D ribofuranols. These structures shown here are called the Haworth projection. The 5-membered ring or the 6-membered ring, flat ring, is called the Haworth projection, and it's used in carbohydrate chemistry. Let's take a look at one more example here for D-glucose. Let's follow the exact same procedure as we described just now. First step, you want to lay the molecule down, carbon 1 on the right-hand side. Keep the stereo centers. OH on the right side, in Fischer projection, they will be pointing down. The one on the left side will be pointing up. Next, curl the molecule up. Put your carbon 1 on the right-hand side and curl it up, and curl the rest of the carbon chain up. Here is what we have. Next, we are going to react the carbon 5 OH with carbon 1. Carbon 5 OH is pointing down now, so we need to rotate a carbon-carbon bond to make carbon 5 OH pointing to the right side, close to carbon 1 for the reaction. The bond we are going to rotate here is the carbon 4-5 bond. Okay, so after the rotation, carbon 5 OH is pointing to the right-hand side. At the same time, carbon 6 CH2OH will be pointing up. Now we can have the reaction. Carbon 5 OH form a bond with carbon 1. And here we have two forms or two products depending on the stereochemistry on carbon 1, right? We have the alpha and the beta form. So these are the two cyclic hemiacetal for D-glucose. And how do we name them? Since it's 6-membered ring, they are pyranols, right? So we have beta D-glucopyranols and alpha D-glucopyranols. Again, these are also the Haworth projection in the 6-membered ring form. All right. So now it's time for you to do some practice. Let's try to draw D-fructose in the cyclic form. I want you to draw the structure of alpha D-fructofuranols and beta D-fructofuranols. Furanols, 5-membered rings. Okay, so practice on this, and we will check the answer in class. Now let's summarize how to draw Haworth projection from the Fischer projection. The ring oxygen will be at the back and the anomeric carbon at the right. So we only have two forms, the 5-membered ring or the 6-membered ring. And what's shown here will be the only forms for Haworth projection. If the substituents are on the right in Fischer projection, they will be pointing down. On the left in Fischer projection, they will be pointing up in Haworth projection. And for the terminal CH2OH, I don't know if you have noticed so far, we used all D sugars as example, and we found that the terminal CH2OH after the bond rotation will always be pointing up. So when that CH2OH is up, it tells you the sugar is the D sugar. And when that CH2OH is pointing down, that's actually for the L sugar. The OH on the anomeric carbon are when it's on the same side as the CH2OH on the last chiral center, that's the beta form. Or when it's on the opposite side as CH2OH, it's the alpha form. So the alpha or beta, it's all relative. But just now we've mentioned that the terminal CH2OH, when we have D sugar, it's going to always be pointing up, right? So for D sugars, CH2OH always up, then if the OH on the anomeric carbon is up, that's the beta form. Down, that's the alpha form. Okay, this is true only for D sugar. For the L sugar, it will be the opposite. Lastly, 6-membered ring is called a pyranose. 5-membered ring is called furanose. All right. When the alpha or the beta configuration doesn't matter, you don't have to show if the OH is up or down, then you can use a wavy line in horizontal position, like shown here, to indicate that it could be either alpha or beta form. All right.